All right, so now we're going to talk about sarcomere anatomy. So what we're looking at here, again, this is a myofibril. So these are the cylindrical organelles that comprise 50 to 80% of the inside of a skeletal muscle cell. Remember, myofibrils are made up of those myofilaments, with the most abundant myofilaments being the actin and myosin. Those are the contractile myofilaments. Remember, myosin are the thick filaments. They have those globular heads. Actin are the thin filaments. And on those thin filaments of actin, you find the regulatory proteins called troponin and tropomyosin. Some other protein structures that you see in a sarcomere. So now we're going to talk about the anatomy of it all along the length of these myofibrils. These myofilaments are arranged in sarcomeres. Think of sarcomere as the functional unit of a skeletal muscle cell. So we're going to talk about its anatomy, its arrangement. Some other proteins that you find, these are those structural proteins. So let me just delineate what a sarcomere is first, and I'll talk about my first structural protein. So from Z disc, you can also call it a Z line, either or. From Z line to Z line is one sarcomere. And then there would be this Z line to the next Z line would be another sarcomere. And then from that Z line to the next Z line is another sarcomere. So those are the repeating units. From Z line or Z disc to Z disc is one sarcomere. And the Z discs are made up of, so down here, this is just a histological image of showing you what it would look like on a microscope versus the cartoon image. That's just a little easier to digest first. What the Z disc is, it's made up of structural proteins that if you look at what is anchored to them, so the Z disc, these proteins anchor the actin filaments. Okay, and they also attach this other structural protein called titin. So this spring-like coiled structural protein is titin. All right, so titin. So what does the Z disc do? These are it's made up of structural proteins that anchor actin, as well as attach titin. What does titin do? We'll look at its shape. It's a spring-like coiled protein which attaches to myosin, the thick filaments. And so titin anchors the thick filaments, these myofilaments, to the Z disc as well. All right? And then another set of structural proteins are straight down the middle of this sarcomere. This is called the M line, and the M line is also made up of structural proteins. And if you look, they help anchor and they form cross links between the myosin. So these are all structural proteins that help hold these myofilaments in place, stabilize them so that as they're continuously contracting and relaxing, they're stable and maintain their shape. Okay? All right, so those are the structural proteins that I didn't really show in the last video, but I just did. All right, so let's talk about the more anatomy of the sarcomere. I'm actually going to I added this little simplistic diagram for simplicity's sake, and it's a little bit easier to see and to know the parts first, okay? All right, so thick filaments, these are myosin with the globular heads. These are thin filaments, just the little straight lines. M-line, structural proteins that cross-link myosin. Not shown here would be titin. This is a Z disc. So from Z disc to Z disc is one sarcomere. All right, so let's talk about different bands. And these bands are what we see when we look at a histology slide of a skeletal muscle cell. We can see alternating light and dark bands. That's what we get, that's those striations. And it's because of this arrangement that we see those striations. All right, first, I band. The I band is where you see only thin filaments. So right here, where you only see thin filaments, this is your I band. So it would appear light on the microscope, okay? It's your I band. Now looking at the center, here's our M line. your H band or your H zone, you can call it either or, if you look from here to here, what do you only see? From here to here, you only see thick filaments. Right? So an H band is only thick or only myosin. A bands are from here to here, and what do we see from here to here? Overlapping thick and thin. Overlapping thick and thin. I band, only actin, only thin. Okay? H band, only thick, only myosin. A band, overlapping thick and thin filaments. All right, this is all listed here. You can read this on your own if you didn't understand what I just said. I like this picture. This is just written out what I just went over pretty much. So this is showing you, so here's our myofibril cylindrical organelle, and I like this little cartoon just because it shows you it's taking a cross section through each of the bands. And so here they are color coded in real life. No, your myosin is not red and your actin is not purple, but for these cartoon images, it's easier to make them different colors. But I like this because it's taking a cross section through the I band. What's the I band? Only actin. So we see only actin, only those purple myofilaments here. Then we take a cross section through the H zone and we see only thick filaments, only myosin, which is in red. And then we take a cross section through the A band, all right, through the A band. And what do we see? We see both actin and myosin because they're overlapping. And so this is the A band. So I like this little cartoon just to really get the point home. Okay. Let's see. Does it might, okay. I'm going to skip ahead just to this picture right here, and I'll highlight it in a second. But remember I talked about in my previous video that these globular heads on myosin attach to the actin both above and below. And then they're hinges, and so they pivot in towards the M line. And so these heads will pivot in towards the M line this direction. These heads will pivot in towards the M line in this direction. And what ends up happening is when a skeletal muscle contracts, the actin myofilaments slide past the myosin in towards the M line. And that's what we call the sliding filament theory, all right? So if we look up here, this is a relaxed sarcomere. And I know it's relaxed because I see a prominent H zone, which remember H zone is only thick, only myosin. And I see pretty large I bands, which are only actin, only thin, okay? Now down here, I'll see that this is a contracted sarcomere, and I know that it's contracted because the H zone is now so small. It's pretty much just an M line. All right, so very, very small H zone, and the I bands have also shrunk. And the reason for that is because the actin has slid in towards the M line. The actin has slid in towards the M line. So actin slides past the myosin because those myosin heads attach and pivot, attach and pivot, attach and pivot, okay? And so the actin slides in towards the M line past the myosin, shortening the sarcomere. This is happening along the entire length of all of those myofibrils, and so the entire cell will shorten or contract, okay? And what you see here is a more prominent A. So the A band, you see lots of just overlapping thick and thin filaments. The I bands have shortened, and the H zone has pretty much disappeared, okay? All right, so let me go back to, all right. So I skipped ahead a little bit. That's okay. All right. So skeletal muscle organization. So I just talked about the anatomy of a sarcomere. And then let's talk about, okay, so organization of a skeletal muscle organ. So let's talk about the connective tissue investments. Here's that picture we looked at in the previous video. Here's a bone. Now, this connective tissue is called a tendon. So this would be made up of that dense, regular connective tissue. Most tendons are going to be kind of a rope-like structure, which will attach a skeletal muscle organ to a bone. That's what a tendon does, attaches muscle to bone. Now, the collagen fibers of the tendon will intertwine with a connective tissue covering called the muscle fascia. If you've ever heard the term fascia, like if you've ever, cleaned a chicken or meat, and there's all this kind of feathery, I say feathery if that makes sense, but this white connective tissue that very easily rips off when you're taking skin off of something, right? That connective tissue that rips pretty easily, that's fascia, and it helps kind of bind organs together. It's primarily loose, areolar type connective tissue. There's subcutaneous fascia, which would be the connective tissue right below the subcutaneous layer of the skin, and that would actually intertwine with the muscle fascia, and that helps anchor skin to muscles as well. All right, and then continuous with this muscle fascia would be a connective tissue layer which is directly adhered to the skeletal muscle organ itself. This is called the epimysium. Mysium referring to muscle, epi meaning on the outside or above. So around, directly adhered to the outer skeletal muscle organ. It surrounds the organ. That's epimysium. It's a connective tissue investment, so it's going to be vascularized. It's going to have nervous tissue, okay? Cross-section through the skeletal muscle organ, you'll see that it's made up of bundles. These bundles are called fascicles. Each fascicle contains multiple skeletal muscle cells. The entire fascicle is surrounded by a connective tissue investment called the perimysium. Mysium meaning muscle, peri, think like perimeter, but this is around a fascicle. What is a fascicle? It is a bundle of skeletal muscle cells. All right, this perimysium will also be vascularized. It will also have a nervous tissue supply. And then pulled out from this fascicle is an individual skeletal muscle cell. So here it is, enlarged. I already talked about the connective tissue investment that surrounds a skeletal muscle cell. That's the endomysium. It's a connective tissue investment which surrounds an individual skeletal muscle cell. All right? And what this is showing you is it's pulling out. So you see lots of cylindrical type structures when you're looking at skeletal muscles. Don't get confused. This is the skeletal muscle cell. These are myofibrils. They are organelles within a skeletal muscle cell. This is not a cell. This entire structure is a cell. Then the myofibrils are composed of those myofilaments, actin and myosin, arranged in sarcomeres. So the myofibrils themselves are arranged in sarcomeres, and we already talked about that anatomy previously. This is just talking, okay, so since we just talked about the connective tissue investments, this is a clinical application of understanding those connective tissues. So by the way, why do we have these multiple connective tissue investments? Why did I just spend time talking about it? Well, if you remember, I mentioned several times that the connective tissue investments are vascularized. They also contain nervous tissue supply because we have to have some sort of nervous input for muscle contraction. So it supplies all of that, and having multiple connective tissue investments throughout the entire muscle. Skeletal muscles are very metabolically active. They require lots of oxygen because they contract a lot. They need lots of ATP energy, so they need lots of oxygen to make that ATP energy. So they need lots of blood. They need a very good blood supply, okay? They also need nervous input so that each one of these skeletal muscle cells can contract. And so having multiple connective tissue investments allows for vascularization throughout the entire organ. It also helps somewhat compartmentalize parts of the skeletal muscle so that if there's damage to one area, it's kind of compartmentalized to that area. And it also helps with transferring force from muscle cell to muscle cell. So because there's connective tissue running throughout this connective tissue investments throughout this entire skeletal muscle organ, binding cells together, binding fascicles together, when one contracts, that connective tissue pulls on the other, and it kind of helps transfer force to other muscle cells nearby. So that's the why we have those connective tissue investments. If you've ever heard of Duchenne's muscular dystrophy, this is a degenerative muscular disease exclusively in boys that has to do with the fact that the gene for the protein dystrophin is encoded in the X chromosome. So females have 2 X chromosomes. So if they have a defective dystrophin gene on one X chromosome, well, females have another X chromosome that makes good dystrophin. So it's fine. But in males have XY. And so if they have a defective dystrophin gene on their X chromosome, they don't have another X chromosome to make up for it. They don't have a good copy of dystrophin. They only have the bad. So that's why you have this genetic disease exclusively in boys, okay? What is dystrophin? It's actually a structural protein in striated muscle cells. So striated meaning cardiac muscle and skeletal muscle. Those are the tissues that are going to be affected here. And this dystrophin protein actually anchors the sarcolemma, which is the plasma membrane of muscle cells, to the surrounding connective tissue and to myofibrils. All right, so this protein anchors the plasma membrane of skeletal muscle cells and cardiac muscle cells to the surrounding connective tissue and to myofibrils. You can envision, after we just learned all of that anatomy, that anything that is going to be defective in those muscles are constantly contracting all of the time. These structural proteins are key for maintaining stability. And so if dystrophin, which anchors the plasma membrane to connective tissues and to myofibrils, is defective, a lot of that stability is lost. And so over time, the skeletal muscle, the plasma membrane starts to break down because it's not working properly or it's not stabilized properly. And then that skeletal muscle or that cardiac muscle starts to get replaced by fatty and fibrous connective tissue, which you can envision would be very bad. And that's why it leads to weakness in the proximal limbs, usually wheelchair bound by age 12. You have respiratory, again, that diaphragm is skeletal muscle, and cardiac failure by age 20. So that's why. Am I going to ask about symptoms and stuff? No, but I just think this is a neat application for applying what you just learned about the structure of skeletal muscle cells and myofibrils and the connective tissue investments. All right. And this is just showing you a histology image of healthy muscle fibers and then the Duchenne's muscular dystrophy, where you see muscle cells being replaced by fibrous tissue as well as fatty tissue. So you should be able to, if I give you on the test a sarcomere, you should be a picture of a sarcomere, you should be able to tell me which bands, if I have it labeled, which bands are which, or what is the I band, what is the A band, what is the H zone or the H band. You should be able to tell me about these regulatory proteins, the structural proteins, the contractile proteins. All right, this is all really key to muscle physiology as well. That's all important. Okay. And then this is that sliding filament mechanism or sliding filament theory that I talked about earlier. This is just a concept boost that kind of shows you with fingers so you can envision. This is pretty much what a sarcomere does, is that these actin filaments just slide past myosin in towards the M line. And then this is showing you up here. So you should be able to recognize whether a sarcomere is relaxed or contracted. What's the main thing to look for is the H zone. If it's pretty much gone, if there's little to no H zone, well, then this is a contracted sarcomere. If the I bands have shortened, this is contraction, this is relaxation, okay?